Hi students, we have already entered the lesson, that is what we have the chapter, Applications of Biotechnology. Under this one, the first application in medicines. In that one, we have concluded how far insulin hormone produced by this technology and what is its use. That's the first one. Now the second one, we have to talk about a protein, the one which is found in milk, human milk, that is secreted by women at the time of just birth. And that protein is called human alpha-lactalbumin. This is the protein normally found in human milk, secreted by the mammary glands. Now, it's also having some nutritional value, can be produced through recombinant DNA technology with help of transgenic animals. Now, what is that protein? Now, this protein, what is called alpha-lactalbumin, is composed of 123 amino acids. And the amino acids are actually in the form of chains, which are joined together by means of 4 disulfide bridges, as in the case of insulin hormone or in the case of immunoglobulin molecule. Here there are 4 disulfide bridges. If we are taking immunoglobulin Ig, there are 3 disulfide bridges. 2 disulfide bridges found between the H chain and L chain, and 1 disulfide bridge is found between the 2 heavy chains, and there is a 1 in the case of immunoglobulin molecule. But here the total number of bridges or bonds 4, and the molecular weight of this protein is about 14,178 Dalton. Now if you are just taking the percentage of this protein, it is a most abundant protein in the human milk, comprising nearly about 25% of the total protein found in the milk. That's its value, 25% of total protein found in human milk. I mentioned already it is synthesized by the cells of the mammary glands. Normally, this protein binds with zinc and calcium ions and possesses the properties. What property? And it possesses normally bactericidal activity, having the ability of killing bacteria, defense mechanism, and also anti-tumor activities. So binding with the calcium and zinc and possesses what properties or what activities? One bactericidal activity, another one anti-tumor activity. Now, we have to improve the nutritional value of cow's milk. So, improvement of nutritional value of cow's milk can be done with the help of production of transgenic animals. Now, these transgenic animals can have the ability of producing recombinant human alpha-lactalbumin. Recombinant human alpha-lactalbumin. The attempt has been made to synthesize or to improve the nutritional value of cow's milk with the expression of what is called the transgenic, maybe we can say transgenic animals, to produce or to express what is called the recombinant human alpha-lactalbumin. The attempt was made. Now, scientists just produced healthy transgenic cows by a mechanism. The mechanism is called somatic nuclear transfer. Somatic nuclear transfer. So through this method, you will be studying about the cloning mechanism. The cloning mechanism normally involves, we see that one, somatic nuclear transfer. So by means of somatic nuclear transfer, scientists have produced transgenic cow to increase the nutritional value or to improve the nutritional value of cow's milk, to increase what we have, the human lactalbumin, alpha-lactalbumin in cow's milk. Because in human beings, we have actually 25% of total protein found in human milk is the richest value. The same nutrient content, or to increase the nutrient content in cow's milk, transgenic organisms or transgenic cows were produced. Now let's see what mechanism was adapted, how far it was useful in the past, now also, that is what the alpha-lactalbumin protein. Now we have produced what is called the transgenic cows. What do you mean by transgenic animals? Animals having their own genome along with the genome of some other animals. Okay, such animals are called normally just transgenic animals. So more animals are produced transgenically. Say an example of mice. That's the most abundant transgenic animal. That is mice. Many experiments were conducted and many transgenic mice were produced when compared to other animals. We have produced also fishes, the pigs, all transgenic animals for different qualities. Likewise here, healthy transgenic cows were produced. The mechanism adapted somatic nuclear transfer. The same mechanism is also adapted in cloning. Here what is happening, the nucleus from the somatic cell, the diploid nucleus is removed and then just it is to be injected into the egg without nucleus. So that now the egg possesses diploid nucleus. This is the mechanism adapted. So the nucleus from the somatic cell is transferred to the egg cytoplasm, the one without nucleus, by which one can produce what is called actually an embryo. So creation of embryos by using somatic cells and egg cells, that mechanism is called somatic nuclear transfer. By this mechanism, test cows were produced, in which the expression of what is called the cow to produce the milk up to 1.55 grams per liter of recombinant human alpha-lactalbumin was achieved. So the expression of more, that is proteins, when compared to the normal cow was expressed. Now the value is about 1.55 grams per liter of what is called recombinant human alpha-lactalbumin protein was achieved. The same mechanism was also adapted to produce what is called transgenic goats, in which the expression of human alpha-lactalbumin just actually up to, say an example of 0.1 to 0.9 milligrams per milliliter. This is the value in the case of goat milk. The increased amount of what is called the lactalbumin, human alpha-lactalbumin. Now, one cow was produced in 1957. That is what we have Rosie. In 1957, a cow Rosie, a transgenic cow was produced. That cow produced a milk which contains what we have the human lactalbumin. So it is enriched with human lactalbumin. That's a cow which produced milk enriched with human lactalbumin. Now what is actually this value? What is the importance of the milk produced by this cow, the transgenic cow? Now the nutritional value, what we have the protein enriched milk, having the value of 2.4 grams per liter. This is the amount of what is called the protein lactalbumin that is found in that milk produced by the Rosie cow. And this milk is nutritionally a well-balanced one, a well-balanced food for newborn babies when compared to the normal milk produced by the normal cows. So there is a role of what is called actually transgenic animals in the production of human alpha-lactalbumin. So it is found only in the case of human milk. We are producing the animal milk, that is the case of cows or goats, by means of what is called transgenism method, or by means of recombinant DNA technology method, so that we can incorporate, that is milk protein alpha-lactalbumin in goat's milk also, or in the case of cow's milk. One such cow, Rosie, was developed in 1957, which produced human lactalbumin protein enriched milk, a balanced food for newborn babies. Okay, that's about the second one, human alpha-lactalbumin. Now we have to go for the third one under the applications in medicine. The factor, the blood clotting factor, what we have? 8. You studied hemophilia. Let us discuss now. Okay, the role of blood clotting factor. Under the applications in medicine, now the third product what we obtained by means of recombinant DNA technology or BT, that's biotechnology, is human blood clotting factor 8. Now the clotting of blood is affected by nearly 13 major factors. Factor 1, factor 2, like that factor 13. For example, factor 1 fibrinogen, then factor 2 prothrombin, then factor 3 that is thromboplastin, factor 4 calcium ions, like that. Now factor 8 is otherwise called anti-hemophiliac factor. Anti-hemophiliac factor responsible for clotting. So we have three different types of what we have the hemophilia disorder, what is seen already under the genetics. That is hemophilia A, hemophilia B and hemophilia C. So the bleeders disease. In this case, we know that the blood fails to clot. Now hemophilia A is produced because of the absence of factor 8. Then hemophilia B is actually because of the absence of factor 9. And hemophilia C because of the absence of factor 11. So these three factors when absent, ultimately we have the bleeders disease of different types. Now here we are talking about the clotting factor 8. So I mentioned many factors are required for normal blood clotting, that is 1 to 13. One such factor is factor 8. It is otherwise called anti-hemophiliac factor, AHF. Totally 13 factors are responsible, major factors. Now it is an X-linked product. The gene for the synthesis of this factor, that is what we have, 8, is present in the X chromosome. So it is an X-linked what is called a character. So when there is a genetic defect, so when the gene is present in dominant form, now the factor is produced. When the gene gets mutated to form a recessive gene, that is called the genetic defect, that results in the production of a disease what is called hemophilia A, an X-linked recessive gene disease. This is because of the mutation of a dominant gene into a recessive gene. So there is no production of what we call the factor 8. Ultimately the person suffers from hemophilia. Now it has a number of actually symptoms. The main symptoms are actually prolonged blood clotting time. The clotting time is delayed. Normal clotting time is 3 to 5 minutes, but here it occurs about 30 minutes. By that time, the blood is completely drained, so the individual dies. Then another one also we have bleeding, internal bleeding within the organs, internal bleeding. How they treated this disease in the past? So the factor 8 is isolated, that is from the blood of normal human beings and used for the treatment of the disease. But it has its own disadvantages. A blood from a normal human is actually collected, and from that only we are getting the factor 8, and that is to be administered to a person having the disease hemophilia. So that was a whole method. So in that method we have some disadvantages. Now what are the disadvantages? Then how far now the new method adapted to produce this factor? Two methods adapted currently, recently also we have a new method, and through recombinant DNA technology one can produce what's called the factor 8, and that is being used to treat, that is hemophilia. Now let's see. So in the past, for the treatment of hemophilia, blood was collected from normal human beings. But it has its own disadvantage when you are collecting blood. What are the disadvantages of collecting blood from normal human beings? Number one, requirement of large quantities of blood for this purpose. Because we need only the factors found in just a little concentration of the blood, we need large volume of blood to be collected. Then only you can get a little concentration of what is called hemophilia factor. That's what we have, the factor 8. That's nothing but anti-hemophiliac factor. We cannot say that's hemophiliac factor, anti-hemophiliac factor. Secondly, while transfusion is performed, when the blood is collected from a person and transferred to a person without screening, there is a chance of transmission of infectious diseases like AIDS. These are the two disadvantages that was noticed in the past when treatment was made for this disorder, that is what we have hemophilia A. Now for this one, to overcome this defect, now the hemophilia factor, that is what we can say the factor 8, anti-hemophiliac factor, is produced through recombinant DNA technology, but not in the bacterial cells, but in one animal's ovary or in the kidney cells. Now the name of the animal, Chinese hamster. Chinese hamster. So what is hamster? a rat-like animal, with the pouched cheeks, cheek. This is what's also hamster. So now DNA, recombinant DNA technology has been adapted in the hamster ovary or in the kidney cells of baby hamster, either in the adult ovary or in the cells of baby hamster, particularly the kidney cells. These are all the places where the recombinant technology has been made and through which we are getting what is called the factor VIII and the hemophilia factor. Now most recently we have a mechanism what's called a cell line, a continuous cell line of human origin has been used for the first time to produce what is called human blood clotting factor. This is a modern technique. A cell line technology has been used for the production of what is called this factor VIII for the first time. So that's about the human blood clotting factor VIII, how far it is produced, how far actually it causes a disease when there is a deficiency of that factor. Now we have to go to the next one, interferons. The fourth product obtained through applications of biotechnology in medicine is interferon. So normally the interferons are commonly called antiviral proteins. They are not killing the viruses, but normally just giving signals to the cells, you are going to be attacked, be ready to protect your cell. So they just normally giving signals to the cells in the vicinity to escape from the infection of viruses, but they never kill viruses. So interferons are proteinaceous substance, a glycoprotein. It's a glycoprotein. Antiviral, species-specific substance, attack only specific actually substances of species or specific species attack. And they are produced in the mammalian cells when the cells are infected with the viruses. Mainly you see that when the leukocytes, when infected with the viruses, they produce the interferons. Now they were first reported by Alec Isaacs and Jean Lindemann in 1957. Now they are classified into three types based on the chemical composition: alpha interferons, beta interferons and gamma interferons. Now the symbol for this one, INF, interferon. Now the alpha interferon is produced by the fibroblast cells. The beta interferons are produced by the leukocyte cells. And the gamma interferons are produced by the immune cells of the immune system. That's about the nature of the types of cells. In what way they are working? Now normally these interferons are actually chemical modulators also. They are considered as modulators of the immune system. Now they stimulate the cellular DNA to produce antiviral enzymes. These antiviral enzymes inhibit the replication of viruses, thereby protecting the cells. So it prevents the replication of viral DNA, so it prevents the multiplication of the viral, that is what is called the nucleic acid, thereby protecting the cells. Then, so like factor VIII, we can also treat these what we call specific disorders by using the interferons, by collecting interferons from the blood of normal persons. Here also some disadvantage. For collecting the interferons from the blood, it needs large quantities of blood. As in the case of, for example, you see that one, the factor VIII to treat hemophilia. Here also we can collect what is called the interferons from the blood of normal humans, but they need actually large volume of blood. So it is normally unusual. That's one of the disadvantages. To overcome this only, we are having what is called the recombinant DNA technology to produce the factor, that is interferon. Let's see now. Like what we have the factor VIII, interferons can also be collected from the blood to treat the diseases, but it needs large quantities of blood. It is impracticable. So that it's not practicable. So that to overcome this issue, now we are following the new technology, what is called the recombinant DNA technology, to produce what we call the interferons. Now, the host cell for the production of these interferons, unlike others, only the yeast Saccharomyces cerevisiae and not E. coli. For the production of insulin hormone, for the production of growth hormone, we can use E. coli. But for the production of interferons, we cannot use E. coli as the host cell for gene cloning or for multiplication. We need only the E. Saccharomyces cerevisiae. What is the reason? The E. coli does not have the machinery for the glycylation of what is called the proteins. So it is a glycoprotein. Interferons are glycoproteins, one need question. So glycylation to proteins only resulted what we have the interferons. So E. coli does not have the machinery for the glycylation of proteins. That's why another host, an alternative host, namely yeast, has been used. Now, what is the use of this interferon? The interferons are used for a variety of or treatment of a variety of diseases, like what we call the cancer, then multiple sclerosis, one of the what is called autoimmune disease, where we have the hardening of the myelin sheath in the nerve fibers, so that there is no conduction of nerve impulses properly. That is called multiple sclerosis, an example for autoimmune disease. Then hepatitis C. We have hepatitis A, hepatitis B, hepatitis C, then non-hepatitis A, non-hepatitis C, and also like that. There are many types of hepatitis viruses. So here hepatitis C that can be treated with the help of interferons. Then herpes zoster, a kind of, I mentioned about a chickenpox, a viral infection, AIDS, etc. So for the treatment of all these diseases we can use, that is interferons. It's also considered as a modulator of the immune system. Now I discuss about what is called the rosine, the first transgenic cow that was produced in the year 1997, not 1957, it is 1997. So this is also a neat question, and this is also a neat question. This is a glycoprotein, what we have the interferons, and about the nature, the three types, etc. So we have some more applications in medicine by what we have, the biotechnology. Let's see in the next class. Okay, thank you.